Welcome to the deep dive, where we take complex sources and really distill them down for you.
Giving you that essential insight without all the heavy lifting.
Exactly. Today, we're focusing on a really critical environmental issue, invasive species,
hmm
and specifically how we track them in places like rivers, which are, constantly moving and changing.
It's a real challenge.
Yeah, so we're diving into this detailed study that tries to combine two pretty cutting-edge techniques. We're talking genetic sampling and aerial surveillance.
Right. Trying to solve that big question. Can we actually measure how much of an invasive plant is out there?
Not just if it's there, but the quantity.
Precisely. The paper we're looking at is titled Broad-Scale Detection of Environmental DNA for an Invasive Macrophyte and the Relationship Between DNA Concentration and Coverage in Rivers. It's led by Hiroyuki Doi and colleagues.
Okay.
And it really digs into whether environmental DNA, that's eDNA, can go beyond just presence-absence detection.
Can it tell us about biomass in a flowing river system?
That's the core question.
So our mission here is to unpack what this eDNA approach can do for rooted plants, things stuck to the bottom, especially when you compare it directly with high-res drone photos, UAV photos.
Unmanned aerial vehicles. What does each tool tell us? Can they work together effectively? That's what we need to figure out.
Okay, let's set the scene. What's the specific problem here?
Well, the study focuses on invasive submerged macrophytes. These are aquatic plants living fully underwater.
Right.
And when they invade, they can cause, well, huge disruptions. They outcompete native plants, sure, but they also change the whole ecosystem.
How so?
They alter water chemistry, they change how the water flows, they can really choke out habitats for fish and other organisms. It's fundamental change.
And the specific plant in this study is Egeria densa, Brazilian waterweed.
That's the one. It's known for being really aggressive, forming these super dense underwater meadows. It's a global problem, really, but particularly bad in parts of Europe and East Asia.
And the paper looks at Japan specifically.
Yes. It zeroes in on Japan, where E. densa has a long history. It was introduced way back in the 1910s, believe it or not.
Wow, over a century ago.
Yeah, initially for experimental stuff. But now, it's a massive ecological and economic headache. It forms these thick mats on the riverbed.
Impenetrable mats, the paper says.
Exactly, which means huge problems for people using the river.
Yeah, this isn't just an aesthetic issue, is it? It's about real functional problems.
Absolutely. These mats block waterways, they seriously hinder fishing activities, they stop boats.
Which must cost a fortune to manage.
It does. Water authorities spend a significant amount each year just clearing this stuff out, physically removing tons and tons of plant biomass.
So, okay, the big challenge then is knowing where it is and how much there is, right?
Right.
Measurement.
Precisely. Because the old ways, sending divers down, dragging grappling hooks, direct observation, they're incredibly expensive and time-consuming.
And you just can't scale that up for a whole river basin. It's impossible.
No way. You need faster, broader techniques.
Which brings us to the two solutions this paper tests. First, the aerial view, UAV photos.
Right, drones. They've been used more recently to estimate how much area these plants cover on the riverbed, basically mapping the cover percentage.
And the second solution.
The genetic approach. eDNA, collecting DNA fragments directly from water samples.
We hear a lot about eDNA for fish and amphibians.
Exactly. It's been revolutionary for detecting mobile species because you find their DNA without ever seeing or catching the animal itself.
But the big question here is, does that translate well to stationary plants, things rooted in place?
That's the million-dollar question the study tackles.
Okay, so how did they actually go about testing this? What was the setup?
They conducted field surveys in two different river systems in Japan, the Sava River and the Takatsu River. And they chose 12 sites in each river.
12 spots per river. And what did they do at each site?
Well, the sampling was pretty intensive. At each of the 12 sites, they collected just 1 liter of river water.
1 liter. That's it. Seems tiny for a whole river section.
It does, but that's the power of eDNA sensitivity. They filtered that liter right away to capture any suspended DNA material.
And then they analyzed that filtered material.
Yes, using quantitative PCR, qPCR. This technique doesn't just detect DNA. It counts the number of copies of a specific target sequence.
So they could quantify the Egeria densa DNA.
Exactly. They used a primer and probe set designed only to find and count DNA from that specific invasive species.
And they were careful about contamination. That's always a worry with eDNA.
Extremely careful. They ran multiple negative controls, things like cooler blanks, which is sterile water kept with the samples, and equipment blanks, where they rinse their gear with sterile water.
To make sure any E-densa DNA they found actually came from the river?
Precisely. Rigorous quality control.
Okay, so that's the eDNA side. What about the drones, the UAV part?
They used DJI Phantom 3 drones, flying them at a standard altitude of 100 meters over the sites.
Capturing photos from above.
Right. These photos were then processed using GIS software, ArcGIS, specifically to calculate the percentage of the riverbed covered by macrophytes, by the plants.
And they didn't just look right at the sampling spot.
No, and this is really important, they calculated this plant cover percentage at multiple distances upstream from where they took the water sample.
Upstream? Why upstream?
Because the water flows downstream, carrying the DNA with it. They wanted to see how far upstream the plant beds were influencing the DNA concentration at the sampling point.
Ah, okay. How far upstream did they look?
They measured cover at 50 meters, 100, 200, 400, 800, and all the way up to 1,600 meters upstream.
Wow, over 1.5 kilometers, trying to capture that downstream drift effect.
Exactly. And they also added a seasonal comparison.
Right, you mentioned that. Summer and winter.
Yep. The main sampling was in summer, July, August, when the plant biomass is typically at its peak. But they also did a winter survey in November in the Akamatsu River, when the plant naturally dies back.
To see if the eDNA signal dropped along with the biomass? Makes
Yeah.
sense. Okay, let's get to the results. This is where it gets interesting, right? What was the main finding about just detecting the plant, the distribution?
Well, the detection was incredibly successful. They found Egeria densa eDNA in all the study sites, every single one.
Even in sites where the drone saw no plants, 0% cover.
Yes. Even where the UAV photos showed absolutely no local plant cover, the eDNA test was positive.
So that strongly suggests the DNA is widespread throughout the river.
Absolutely. It confirms eDNA is fantastic for just confirming broad-scale presence. If it's anywhere in the system, eDNA seems likely to pick it up.
Okay, great for early warning then. But back to the main goal, quantification. Did the amount of eDNA, the concentration, match the amount of plant cover seen by the drones in the summer?
This is where it gets complicated. There was a statistically positive correlation between the eDNA concentration and the macrophyte cover measured upstream, especially at those distances from 100 meters out to 1,600 meters.
I sense a butt coming.
But the relationship was weak. Really weak, actually. The R-squared values, which tell you how much variation in cover is explained by eDNA. They only range from 0.17 to 0.34.
Wait, hold on. 0.17 to 0.34. That means, at best, the eDNA amount explained only about a third of the difference in plant cover.
That's right. And at worst, only 17%.
So the quantification didn't really work that well. If you're trying to predict biomass from DNA, that seems not great.
It's definitely not a strong predictive relationship.
Yeah.
And it gets even more telling.
How so?
The authors did further analysis. They found that if they removed the sites that had zero plant cover, but still had positive eDNA, and also removed the sites with the very highest average eDNA readings, the statistical significance of that weak correlation, it just disappeared.
So the correlation was basically just driven by presence versus absence and maybe a few hot spots, not a smooth relationship between amount of plant and amount of DNA.
That's what it looks like. And here's the real kicker. You mentioned it earlier. The relationship completely broke down once the plant cover got above about 5%.
5% cover, that's tiny.
It is. Above that very low threshold, it seems the river just becomes saturated with E-densa DNA. The eDNA signal couldn't distinguish between, say, 10% cover and 50% cover. It was just high.
Wow, okay. And did the winter data support this lack of quantitative signal?
It did. In the November sampling, when the plant biomass was lower, there was no significant correlation at all between eDNA concentration and cover, except for one odd negative relationship way upstream at 1,600 meters.
So pretty much confirmed. eDNA detects presence everywhere, but struggles to quantify local biomass, especially once the plant is even moderately established.
That's the paradox they found.
So why? Why is the DNA everywhere, even with no local plants, and why is the correlation with biomass so poor?
It really comes down to the river's flow dynamics. Think about it. When you study, say, a fish using eDNA, the DNA is shed relatively close to where the fish is, or was recently. But with a plant rooted to the spot, the main source of DNA isn't just leakage. It's physical bits breaking off leaves, stems, cells.
Detached material.
Exactly. It becomes particulate organic matter, or POM. And once that stuff breaks off,
the river just carries it away.
like a conveyor belt, but maybe more like a blender too. It breaks down, disperses, and drifts downstream. This constant shedding and downstream transport effectively smears the DNA signal across large sections of the river.
Homogenizing it.
Yeah, homogenizing the concentration over potentially kilometers. So the DNA you collect at one point might reflect plants quite far upstream.
Which masks the signal from the plants right near the sampling site.
Precisely. The broad-scale distribution signal swamps the local quantity signal.
Okay, so what does this mean practically for the people trying to manage these invasive weeds?
Well, it means eDNA is still incredibly useful, but maybe not for the reason they initially hoped. It's fantastic for broad-scale surveys of presence or distribution.
Like mapping which river segments or catchments have the invader.
Exactly. It's cheaper, faster, and requires less effort than traditional surveys. And a big plus, you don't need specialized taxonomic skills to identify the plant underwater. The DNA test does the ID.
So it's a great screening tool, an early warning system.
A phenomenal one. But if your goal is to know exactly where the dense patches are for targeted removal,
Then the eDNA alone isn't enough. You still need something like the UAV photos.
that seems to be the case based on this study. The UAV gives you that crucial spatial precision for local density or biomass.
So use eDNA for the wide area search, find the positive zones, then use UAVs for the detailed mapping within those zones to guide cleanup crews.
That looks like the most effective strategy right now, combining the strengths of both.
Did the study suggest ways to make the UAV side even better?
Yes, they did. They acknowledged that their UAV analysis used standard RGB cameras, red, green, blue light, like your phone camera. And that's subject to things like lighting conditions, water clarity.
Makes sense. Hard to see underwater plants clearly sometimes.
Right, so they suggest moving towards more advanced sensors on the drones, specifically multispectral sensors.
Multispectral, what does that add?
These sensors capture light beyond just the visible spectrum. Crucially, they capture near-infrared light, or NIR,
Okay, and why is NIR important for plants?
because healthy vegetation reflects NIR very strongly. By measuring the difference between reflected red light and reflected NIR light, you can calculate things called vegetation indices. The most common one is NDVI, Normalized Difference Vegetation Index.
NDVI, I've heard of that, used in agriculture a lot.
Widely used, it gives a much more direct and robust measure of plant health and density, essentially biomass, than just looking at color in an RGB photo. It cuts through some of the noise from water conditions.
So using NDVI from drone sensors could make the ground truth part of the comparison much more accurate.
Exactly. It would strengthen that side of the equation significantly.
What about future directions for the eDNA side? Where does the research go next?
Well, one obvious thing is scale. This study looked up to 1,600 meters upstream. But given how important flow seems to be,
You need to look even further. Multiple kilometers.
possibly, understanding the transport distance is key. Also, this study focused on one very abundant, dense invader. How well does this work for rarer macrophyte species? We need more evaluation there.
Good point.
And then there's the potential for eDNA metabarcoding.
Instead of looking for just one species, you sequence everything.
Right. It's already being used effectively for surveying whole communities of fish or invertebrates from water samples. Applying that to macrophytes could let managers monitor all the plants invasive and native in a river section simultaneously.
That would be powerful. A whole community snapshot from one sample.
Huge potential there.
So wrapping up, the big takeaway seems to be that river flow itself is a massive factor. The fact that a single water sample can detect a plant kilometers away because its debris drifts downstream, that really changes how we have to think about monitoring rivers, doesn't it?
It absolutely does. It highlights the interconnectedness, the downstream transport. What happens upstream definitely impacts what you find downstream, maybe more directly or over longer distances than we sometimes assume for eDNA.
Which leads to a final thought for you, the listener. If eDNA is this great, cheap tool for telling you an invasive plant is around, maybe far upstream, but you need expensive, high-res aerial data like UAVs with fancy sensors to know exactly where to send the removal crews.
How do you make that practical decision?
Right.
Where's the balance point? How much confidence do you need from the eDNA before you invest in the detailed mapping? That cost-benefit analysis, balancing broad detection against targeted action, that's the real-world puzzle this research leaves us with.